Good morning, everybody. It's Mr. Forshee. Today, I want to go over with you the kind of the beginning of the new unit that we're starting. This is going to deal with Mendelian genetics. This is going to get us ready for kind of the biotech, but also going to get us ready for the cat lab that we'll be running when I get back to school at some point. The thing with this is chapter 4 in your book. It's a good chapter to read. There is an objective sheet I've given you. That'll be in today's folder too. But the notes here just kind of give you a little historical piece of Mendelian genetics. You will have a quiz on Friday. Some of this may be in there. So some to just look over, but I think most of you guys have had some of this before. It's pretty straightforward. Just looks at, dominance and recessiveness and using kind of Mendel's language to show how that pattern works. Now, you can see here, again, with Mendel, we're not going to get into too much, but there's a historical point where you could see this is him right here. He worked as a monk, and one of the jobs he was given, his family had a back history of farming, so they kind of put him in charge of some of the agricultural pieces. And with his curiosity, and again, he had a strong math background, he was trying to figure out the patterns that were developing in, if you were to kind of cross or to pollinate a plant, exactly why that plant looked similar or why sometimes it looked different. Now, to kind of understand the time frame about this is, they didn't think that there was a, gametes, sex cells, but what they thought instead, like with humans, there was something called homunculus, which means that there was a human inside of the sperm cell and it just needed a place to grow. And so that really they didn't think that the female had any hereditary or genetic component for that offspring. And it was coming solely from that of the sperm cell. So you can see this is kind of an example of homunculus look. So just kind of give you a time frame. They didn't know much about this at this point. But, again, Mendel was, after he had published his work, still not quite understood exactly what he was looking at. Now, for a flower here, there's really two things that we can look at. This is referred to as the female sex part of the flower. It's called the carpel. And the stamen is going to be the male section of the flower. So it's the anther and the filament. Now, what happens here is the stigma is the opening to where the egg is actually going to be located inside of that of the ovary. Now, the anther is the part that would have that of the pollen, which is going to be on top of it. So the pollen is really the sperm of the plant. That, again, this would be considered like a complete or whole plant because it can self-fertilize itself. Some plants are not this way. Sometimes we call them incomplete. There's different verbiage on how they call these plants, but I'll just call it complete or incomplete. But this is an example of complete. Now, what Mendel did is he was able to manipulate these plants, and a pea plant is the one that he looked at that was a complete plant. So it had both male and female parts. But what he would actually do is he would cut the sections of the filaments, and then he could remove those, and that's how he can then control the pollination of the plant. So then that's how you could cross different pea plants to look for different characteristics that would happen within the different generations. So you can see here's kind of his his success in terms of being legitimate science or, why pea plants. These are some things we'll look at. But you could see the properties of an experimental organism. So again, it made it, because there's a quick turnaround. It's fairly easy to grow. So it's not like takes all year for one plant to grow. It possesses a number of different traits, and we'll look at some of those today. And self-fertilizing, and again, offspring of self-fertilizing plants should be fully fertile so that the future crosses can be made. So it's a way for them to kind of be able to track exactly if he cross-pollinated it himself, then that plant could eventually self-fertilize itself and carry on to those next generations. So again, we'll look at these, but it's really kind of the perfect plant for him to run. And again, like I said, short growth period for mature plants. So those are just some things that made it successful for him to follow these patterns that we're going to look at today. Now, here are some of the different things here. This is something like for the quiz you might want to think about here. You can take notes on these too on the PowerPoint anytime you want to. But you could see between like the dominant versus recessiveness, like sometimes you would see like the green is dominant to that of the yellow. And then, round was dominant to that of wrinkled, seed color. So there's a number of different traits you could see in terms of, again, how patterns would work. So this is really kind of the dominant traits, and these are going to be the recessive traits. Now, why does that happen? Well, it's just like with us. If we look at purple-white color for that of a pea plant, so if you go like purple to white, if we're looking at flower color here, and that flower color, we could see that if there's a purple, I'm just going to put this in the capital P, and then that for white, I'm going to put that in a lower p. So really the homolog pair here would be big P, little p, which would then give it its purple color. So because of that, because it's a homolog pair, so one's got to come from the anther, and then one's got to come from the egg. Again, when that fertilization takes place, we go from that of a haploid cells to that of a diploid cell. So I kind of wanted to show you kind of how it works in terms of the patterns, much like we look at ourselves. If we look at like cystic fibrosis, making a protein or not making a protein, it kind of goes along that of the same lines. Now, what he kind of concluded from his first crosses that he was able to look at, because again, this was kind of the main focus of his crosses, because not everything that he tried worked out very well. So there could have been other factors, something called linkage that we'll talk about down the road. But one of the things that he could come up with was these 3 different postulates right here. And what he considered was that genetic traits are controlled by unit factors that exist in pairs. So that's why, like we said with the plant, you had a big P, little p. So there's kind of existing pairs that control traits. When 2 unlike unit factors responsible for a single trait are present in a single individual, one is dominant while the other is recessive. Now, we can talk about that as being a functioning, non-functioning, but Mendel's language was one thing kind of dominates or overshadows that of the other. So think of that like a tall and short plant. And then the last pair is separate or segregated randomly. So what this was meaning that the process of these plants kind of reshuffling their characteristics were really based on that of being random. And that can happen because a lot of these are going to be on separate chromosomes. So it allows them to have that thing called independent assortment we had talked about before. So here's like a good example of like Mendelian genetics. This is the one we've talked about before, in that we have a mutation. Now this is a dominant trait. So if we have this, we're going to go big A, little a. These individuals here are going to be the ones that have that disease. So this male right here, this male right here, and this female, those are the ones that all have that particular trait. Now, with achondroplasia, it's pretty unique because you can't be homozygous. You have to be heterozygous. For some reason, a homozygous combination of those genes will cause the embryo to spontaneously abort. Now, if you notice the ones in the back here, we have 3 other individuals. Because they have normal stature, they're going to be like little a, little a, little a, little a. Okay? So we can kind of see he's going to be little a, little a. So again, but we can use Mendel's kind of understanding of how these genes travel from generation to generation, make some simple predictions. Now, achondroplasia is not with humans. So if you look at corgis, dachshunds, different types of even horses, chickens, they all kind of display this achondroplasia. So it's not just within humans, but it can be in other animals. We can see these particular conditions. Now, how do you do this is by using kind of Mendel's idea of using something called the Punnett square. Now, you guys have done Punnett squares before, but you could see, like, here's an example. If we go back and we look at the two parents that we had back here, both of them mom and dad. So we could say this is going to be dad, and this is going to be mom. Again, from with the chondroplasia. If you notice here, that AA is going to be lethal. So we have really a 1/3 is going to be normal, and 2/3 will have dwarfism. But you can see how that works in terms of kind of tracking down. Now, again, you guys have done this before, but it's simply all we're going to do is we're going to put down, for example, this letter goes here, this letter goes here, this letter goes here, this letter goes here, and so forth. So in terms of kind of understanding how this works, this is where we can talk about something like this would be, again, when we talk about genotype and phenotype, this would be for the, like the phenotype. And then the genotype would be, we would have 0 out of 4, we would have is big A, big A. Then we're going to have 2 out of 4. Now, let me correct that. I'll just write this down because even though it is lethal, let me redo that. This one would be 1/4 big A, big A. Then it'd be 2/4 big A, little a, and then 1/4 little a, little a. And this would be the genotype. So depending on what I'm asking you guys down the road, this is where you guys can write that in. Sorry about that little bit of a mess up. Yeah, you still recognize it as a genotype, even though it doesn't happen. There is a possibility, but the likelihood of that happening is not going to be too significant. Now, in terms of understanding how this works, and this is where some of you guys are some a little bit of trouble, I think, with that of meiosis, is something called the law of segregation, and derives from the fact that during meiosis, alleles separate into different gametes. Well, what that means exactly is like when you have duplicated chromosomes, when they separate, they each go into their own gamete. So remember with a male, we start off as one diploid cell, but we go to 4 haploid cells. Those 4 haploid cells are going to have different combinations of genes in there depending on terms of how they line up. So again, that's just going to be called the law of segregation. So the law of segregation means what we do is we have different variation of how things can show up. So if you look at here, this is kind of an example of like Mendel's data that he had collected over the years. So he looked at like seed form, for example, and he collected 7,324, and then of that, dominant were 5, 4, 7, 4, and then 1850 for recessive. So that ratio came out to be 2.96 to 1. So if you notice that all these, they all are almost like a 3-to-1 ratio. So that's one of the things you could look at again in terms of with, there's certain patterns that show up. Now, the more number values you have, the better your number values will be. But you could see that Mendel kept doing this over and over, keep seeing these repeating values show up. Sometimes he had a 1-to-1 ratio. So he knew that something had been going on that's different from his experiments from previous plants that he had looked at. So if you look at this right here, this is kind of an example of what we're going to look to do. And we can kind of see how this works within that of the male with